You have a Zoom? Zoom? Yeah. Okay. Thank God. Done? Okay, right. So right now, we are coming to actually, this is the last step, the Krebs cycle, step 3. But step 3 is the last step of respiration. But there's another step coming up, step 4, which is not directly related to this. So I write here step 3. Krebs cycle, which is also take place in matrix. Right. Again, Krebs cycle is it has a many steps. Krebs cycle has many steps, but you don't want to remember any steps and nothing to remember all the steps like this, just to get an idea. For example, this is the reaction 1. Uh, Oxaloacetate, citrate, isocitrate, succinyl-CoA, succinate, fumarate, malate, like this. So there are many reactions take place. And I will show you how exactly it's happening. Now, this is the acetyl-CoA, and it has this 2-carbon acetyl group from pyruvate. This is a CoA. So acetyl-CoA, which is formed in the link reaction. So acetyl-CoA donate this 2-carbon compound to the Krebs cycle. The Krebs cycle, the name given because the person discovered this cycle, Hensley and Krebs, they give this name Krebs cycle, right? So 2-carbon acetate group is donated to the Krebs cycle. The starting compound of Krebs cycle is 4-carbon. So there's a 4-carbon compound which is going to join with 2-carbon compound. So then it can form 6-carbon compound. So 6-carbon compound, they are going to break down in a series of reaction again to come back to 4-carbon compound. So what happened to these 2-carbon then? So 6-carbon return back to 4-carbon, return to 4-carbon. So what's happened to the 2-carbon now? Okay, here you will see one carbon released as carbon dioxide. So then it can make again 5 carbon compound. And another place, another carbon dioxide is released to make 4 carbon. So 2 carbon dioxide molecule is released. Now you can understand if I if I if I show you this is a 1 carbon dioxide molecule, this is a 1 carbon dioxide molecule, and this is 1 carbon dioxide molecule. Now pyruvate has 3 carbon. The pyruvate molecule is completely break down because 3 carbon release. So carbon release, 1 carbon release here, the second carbon release here, and third carbon release here. So pyruvate has 3 carbon, all 3 carbon release. And the cycle continue. So we are not going to learn everything in the cycle. We're going to learn only few things. The cycle, when this cycle continue, it can produce 3 things. What are the 3 things? The energy released from this cycle. So they can form at different level 3 times NADH molecules. And they can produce 1 time FADH2 molecule, and they can also produce 1 ATP molecule. So energy is released as 1 ATP molecule, 1 FADH2 molecule, and 3 NADH molecules. So completely energy released, and pyruvate is completely oxidized. So this is the Krebs cycle. 2 carbon acetate completely break down into 2 carbon dioxide, and energy is used up to form 3 times NADH, 1 time FADH, and 1 ATP molecule. So can you quickly copy this down, the Krebs cycle? Okay, Krebs cycle again, acetyl-CoA donate the acetyl group, and Krebs cycle has a 4-carbon compound, makes 6 carbon back into 4 carbon again. So glucose molecule is completely oxidized now in Krebs cycle. Now we're going to end up this process, and we're going to summarize how many molecules are formed, complete oxidation of glucose molecule. Before that, this is for one pyruvate. So can you remember we told 2 pyruvate molecules are formed. So what I showed is for one pyruvate molecules. Then the second pyruvate molecule also enter into the cycle, and it goes to the link reaction. It produces NADH, and it goes to the Krebs cycle. It produces again 3 NADH, 1 FADH, and 1 ATP. So then the pyruvate molecules, 2 pyruvate molecules. So 2 pyruvate molecules enter into the link reaction and Krebs cycle. So the link reaction, they can produce 2 NADH. Krebs cycle, 6 NADH, 2 FADH, and 2 ATP molecule. So what I showed here for one ATP molecule, one pyruvate molecules, when we have a 2 pyruvate molecule, it can produce 2 NADH in link reaction and 6 NADH and 2 FADH and 2 ATP in Krebs cycle. So this is from Krebs cycle, and this is for link reaction. And glycolysis, it produces 2 NADH and 2 ATP. That's from glycolysis. So now a total 2 plus 2, 4 ATP molecule, 2 plus 6, 8 NADH molecule, no more than that, right? So 6 plus 8 plus 2, 10. So this is 10. 10 NADH and 2 FADH. 4 ATP molecules, 10 NADH, 2 FADH2. That's a total molecule produced. Okay, can you also summarize this? The whole energy molecule produced in total oxidation of a glucose molecules. Right now, if you look at the total ATP molecule is 4, which is not much. And it produces more NADH and FADH2 molecules. So these molecules rich in energy. So they are electron carriers and the energy is trapped in NADH, energy is trapped in FADH2. So these molecules now enter into completely different method to get the ATP. So if you want to find why nature did not create a method to completely make ATP at the first harvest, uh, very difficult to find the answer, right? So why complete breakdown of glucose molecule? Why we can't get only ATP? Why we have to ended up with these other molecules like this, and later they're going to make ATP. So that's how it's happening. NADH and FADH, they are rich in energy. They are going to form energy in another completely different method. The second step or second phase, they are going to make ATP from NADH and FADH. So before we come to that, quickly summarize. So we learn glucose molecule in the presence of oxygen produce this much of energy. And ATP is the universal energy currency. So there are steps called phosphorylation and electron carriers. They harvest energy and transfer energy at various levels. Glycolysis is the first step in which glucose breaks down into 2 pyruvate and certain energy is produced, 2 ATP and 2 NADH. The link reaction takes place in matrix. It can only produce NADH and no ATP. Then the Krebs cycle takes acetyl-CoA and completely oxidized to produce NADH, FADH, and ATP. That's a total energy molecule produced. 4 ATP, 10 NADH, and 2 FADH2. So 10 NADH and FADH2 later move into the completely different process called electron transport chain and oxidative phosphorylation. So this is uh, the process in which we're going to see how NADH and FADH2 release the energy to form ATP. How NADH and FADH release the energy to form ATP. ATP. We're going to learn under electron transport and oxidative phosphorylation. And the location is inner mitochondrial membrane. The location is inner mitochondrial membrane. Can you quickly copy this down? The aim of this electron transport chain and oxidative phosphorylation, we're going to learn how NADH and FADH release their energy to form ATP. Okay, you can see here outer mitochondrial membrane and folded inner mitochondrial membrane. The space here is called intermembrane space. Between outer membrane and inner membrane, the space is called intermembrane space. As you know that these membranes are phospholipid bilayers. And phospholipid bilayers also contain proteins which are embedded. Okay, here I am going to show you a complex, a protein complex, which is embedded in the inner mitochondrial membrane. And there's another one and another one. So this protein complex, we can label 1, 2, 3, 4. So I can label them as electron carrier protein. So these are called electron carrier proteins 1, 2, 3, and 4. Okay, we go by step. Maybe you can also draw the one outer membrane and inner membrane, and you can also show these 4 protein complex. So this is the outer membrane. This is the inner membrane. And this is the space, is intermembrane space. And here is a matrix. So these proteins have multifunctions. They can carry electrons. They can act as enzymes. They can also act as a transporter. So they have multifunction, complex proteins. So they could do a electron carrier, enzyme action, and transport. And there's a 2 subunit we can still find here and here. That's another uh, electron carrier proteins complex. 2 additional one, but we mainly focus 1, 2, 3, 4. Okay, so you have a space, second page. So we can write what is exactly happening in complex 1. NADH oxidized by enzyme to form NAD plus, and energy is released as electrons. Okay, you can write in the next page, in complex 1, NADH oxidized by enzymes to form NAD plus, and energy is released as electrons. Hmm? In complex 1, NADH oxidized by enzyme to form NAD plus, and energy is released as electrons. So we can show them in the diagram like this. NADH enzymatically breaks down again into NAD plus, electrons, and hydrogen ion, protons. Now this electron has energy. Okay, then we write in the second one, the electrons, that electrons, right? So I'm reverting this electron, complex 1, complex 3, and complex 4. During this move, energy is lost. The electron move into complex 1, complex 3, complex 4. During this move, energy is lost from the electron. So we can show in the diagram the second step like this. This electron started to move into the complex 1, complex 3, and complex 4, and then goes out. So it does not have energy now. So I don't color it with yellow color because energy is lost. Now we'll see third one. What happened to this energy? The energy of electron are used to pump hydrogen ions in to matrix. Right. Just we're going to finish today's lesson from the point 3, and we can continue from next week. The third point, the energy of electrons are used to pump hydrogen ion into matrix. So I can show it here. So this is number 1, this is number 2, and number 3. Number 3 I can show like this. Hydrogen ion moves in. That is number 3. From intermembrane space to matrix, hydrogen ion pumps in by using this energy. That's the number 3. You can also show in the diagram, and we have to continue from next week. One, two, and 3. One, two, and 3. Okay, so this is end of today's lesson, and we will continue from next week. And if you miss something, if you have any question, please let me know. You can WhatsApp, and we'll see you again next week. Okay, take care. Bye. Videos. A2, Unit 5, Unit 4. Unit 5.